Hey, folks, welcome back. Last segment. So now that we are clear on sort of what happens during the cell cycle, now we're going to look at how do you regulate this stuff. Okay, so there are a couple of interesting experiments done with looking at cell division to help us sort of understand a little bit about what's going on. If we look at number one, remember a cell in S phase. S stands for synthesis, so it's copying the DNA. And then G1 is when the cell is just unduplicated, has chromosomes that are unduplicated, and it's just doing its job, making proteins, doing that kind of business. Well, it happens that if you fuse the membranes of those two cells, the cell that had been in G1 originally will immediately begin copying its DNA and enter S phase. So just by fusing the membranes, you can actually cause a cell to copy its DNA. Even more remarkable is if you look at experiment number 2, you can take a cell that is in cell division. It's actively dividing in this M phase. And if you fuse its membranes with a cell that's in G1, hasn't copied its DNA, hasn't started copying organelles, any of that business, if you copy it, then it will automatically get that cell to begin dividing as well. So how the heck is that? You can take two cells in different stages of the cell cycle, fuse their membranes, and actually get a cell that was in an earlier stage to jump ahead to whatever's going on in the cell that's in a later stage. Turns out there are molecules in the cytoplasm of those cells. And so there must be molecules that are present that help regulate transitions through the cell cycle. So the cell cycle is really a highly regulated sequence of events, and the transitions between those sequences are controlled by different molecules. Okay, so your book mentions this set of checkpoints. So these checkpoints are places where the cell cycle will pause until certain molecules are present, and then it will transition. So there are 3 that we're going to look at here that you definitely want to be familiar with. The first and most important is the G1 checkpoint. The G1 checkpoint is a checkpoint that, if passed, tells the cell, okay, this thing is going to divide. So the G1 checkpoint is the big trigger here, because if a cell goes past the G1 checkpoint, eventually it's going to divide. If a cell doesn't move past the G1 checkpoint, we'll talk about that later. The G2 checkpoint is the last stop before you start cell division. So with the G2 checkpoint, you make sure that the DNA is copied and all the organelles have been copied, and you have the proteins present that you need for the cell to divide. So that's a really important checkpoint, because it's the one that's like, okay, it's time to divide. The third checkpoint we'll look at is this M checkpoint. The M checkpoint happens during metaphase. During this checkpoint, cell division pauses to make sure that all the microtubules have attached to the appropriate kinetochores. So that way you don't have some chromosomes without microtubules connected to the kinetochore. Because if that happens and the cells start to divide early, you're not going to get the sister chromatids separated from one another. So the M phase checkpoint is really important because it ensures that microtubules have attached to the kinetochores on both sister chromatids, so they'll separate evenly. So that's an important checkpoint as well. All right. So going back and talking about that G1 checkpoint, it's a really important checkpoint because, again, if you go past G1, that cell is going to divide. Well, now, what if a cell never goes past the G1 checkpoint? Then it enters a new phase that we haven't referred to yet. It's called G0 or G0. Okay. So the G0 phase basically makes it a non-dividing cell. The cell isn't going to move past G1, so it's just going to go make proteins and do whatever its job may be. We see that with the neurons in our brains. Our neurons will typically be non-dividing cells. So the neuron is made, it's not going to divide again. It just sends electrical impulses. So those are cells that are arrested in G0. Muscle cells, skeletal muscle cells, they'll last like a decade. So those will typically be in G0 as well. Now, can a cell ever revert back from G0 and then basically move past that G1 checkpoint and divide? Yes, cells can be stimulated to do that. So if you can figure out, what combination of molecules to use, you could potentially stimulate cells to divide. Now we're going to look at a couple of molecules that actually cause cells to move past that G2 checkpoint. So this is just one specific, for instance. I think the new curriculum says you don't need to memorize these terms, but they'll ask you stuff related to this. So I think it's helpful to at least recognize how one pair of molecules work together to control one checkpoint in the cell cycle. So what we're looking at are two molecules, cyclins and cyclin-dependent kinases, or CDKs. Okay, cyclin, as its name suggests, it's a protein whose levels will rise and fall. So cyclin levels go up and down in the cell cycle. And CDK, kinase, ACE tells you it's an enzyme. Kin means movement. So it's an enzyme that causes activation of the cell. Okay. But the thing about it is it depends on cyclin. Okay. So CDK levels in the cell stay relatively constant, but cyclin levels go up and down. Now, when cyclin and CDK join together, they're referenced as a new molecule. It's called MPF. It's a maturation promoting factor. I don't know. Have you guys ever seen Power Rangers? Like with the Power Rangers, there's like the Red Ranger and the Green Ranger and the Black Ranger and all that stuff, the Yellow Ranger. I don't remember all of them. So they're individual Power Rangers, but then they join together and then they make this big, like, tiger-looking thing that is even more powerful. Well, cyclin and CDK are different molecules, right? But when they join together, they create MPF, and then MPF can stimulate division of the nucleus. It starts the cell division. So here's another way to sort of view this representation. The purple, darker purple line that you see there is cyclin. So cyclin levels go up, and then they drop. And then cyclin levels go up, and then they drop. And then what happens is when cyclin levels rise, you get more of that MPF superduo, and then the cells divide. When cyclin levels drop, then MPF degrades, and then the cell enters interphase. Then, again, during G2, when you're making these proteins you need for cell division, cyclin levels go up, you get more MPF, and cell division happens. Okay, so again, when cyclin levels change, you get more or less of this MPF molecule, and then the cell either divides or it doesn't. Here's an even better illustration of this. You'll notice CDK, it's the gray or purple. CDK is always present in the cell at constant levels, but cyclin levels will increase, particularly during G2, and then cyclin joins with CDK to form MPF, and then that triggers cell division. When cyclin gets broken down, the cell goes back into interphase. Okay. Let's look at other signals for cell division. Let's see. Now, cells can get internal signals, or stuff from outside the cell can cause cells to divide. So one internal signal, again, this is the signal we were talking about for that M checkpoint. The kinetochores have to be attached to the spindle fiber before anaphase begins. If the kinetochores don't attach to, if the spindle fibers don't attach to kinetochore, then the cell will pause at metaphase until everything's connected before it begins anaphase. A typical external signal are molecules like growth factors. Growth factors, they can dock on proteins on the surface of the cell or inside the cell, and then that will stimulate cells to divide. So here's this PDGF. It's like platelet-derived growth factor. It can dock on these. They're called receptor tyrosine kinase molecules. So this growth factor docks on these proteins. They join together or dimerize, and then that sends a signal inside the cell to begin dividing. So one thing that cells can do is, or one thing that external signals can do, is dock on the surface of a cell, change the shape of the receptor, and then that sends a signal in the cell, hey, you need to divide. Here are a couple of other interesting examples of external signals. One is called density-dependent inhibition. So a lot of times as cells grow, when they make contact with one another, proteins on the surface of the membranes signal to the cells, hey, it's time to stop dividing. So if you get a cut in your skin, you may have sort of a ravine or a canyon in between the two sides of the skin, and the cells will divide until they grow and make contact with one another. And then because of density-dependent inhibition, the cells stop dividing and the cut is healed. So here they show if you grow cells in culture, the cells will grow until they make contact with one another. You can scoop some cells out, and then they'll continue growing until they make contact with one another. Another type of external signal is anchorage dependence. For the most part, cells aren't just like floating around your body. They're held in place to the extracellular matrix. So cells have to be attached to some sort of substrate or surface or material before they will divide. Now, in the case of cancer cells, that's what the CA represents there. Cancer cells may not respond to either of those signals. So even if they get crowded or even if they break loose, they may still continue to divide. So that's, part of the issue caused by cancer. If you have cells that will continue to divide and divide and divide, a lot of times the chromosomes are messed up and the nuclei are messed up in those cells. So they don't work. So they don't do the job they're supposed to do. They just keep dividing. So what you get is a mass of cells that aren't doing their job, and it begins to crowd out cells and organs, so that impairs organ function and can lead to all sorts of problems. In reference to cancer, transformation is what occurs when cells that normally do their job get converted to cancer cells that, again, aren't functioning as they should, but just continue to grow. Now, cancer cells can sort of be in two different versions, right? A benign tumor is a mass of cells that sort of stay put. A lot of times they may be encapsulated. So the cells are dividing out of control, but it's sort of encapsulated and it doesn't spread anywhere. Malignant tumors, on the other hand, again, they do not display any sort of anchorage dependence. They can have pieces of the larger tumor break loose. If it gets in the bloodstream or lymphatic system, they can move to different parts of the body and then sort of take up residence in different organs. So malignant tumors tend to be really problematic because those tumors can break loose, or pieces of the tumors can break loose and then invade other organs of the body. Okay. Here we finish with looking at some images of what type of cell is this, plant or animal? Tick tock, tick tock, tick tock. Good, yes, plant cell, because you see the cell walls surrounding the cells. So here is the kind of stuff I'm looking at. If I look at, say, this cell here, you'll notice there's a nuclear membrane. There's a nucleolus. That's where ribosomes are made. And you don't see any threads there, right? So this is a cell in interphase. Okay. Now, if we look, let's find some decent examples. This prophase, I don't think that's great. But here, I like this better as a prophase. If we look down here, you start to notice that maybe the nuclear membrane isn't as intact. The DNA is condensed into threads. So the DNA is coiling up into chromosomes. So that's an example of prophase. Okay. Metaphase, here's a good metaphase. All the chromosomes have been pulled to the middle, or here the chromosomes have been pulled to the middle. Anaphase, Ana's the action photo, right? So here is a nice example of anaphase where the chromosomes are being pulled apart. Here is an anaphase. This looks like it's getting to be later in anaphase. And then telophase, the dead giveaway for telophase is if you see two nuclear regions. So here, two nuclear regions within the cell, and then you don't see distinct threads anymore, right? That's because the DNA is relaxing and unwinding. So, be able to look at cells. First, be able to tell if it's a plant or animal. Plant, giveaway because it's got a cell wall around it. And then be able to look at cells and figure out, okay, is it interphase, prophase, metaphase, anaphase, or telophase? All right. Well, I hope you're doing well, and this is doing okay for you. If you have any questions, just let me know. We'll get some practice with this stuff, and I'll see you soon. Thanks a lot.